Hello. Welcome, everyone. I appreciate the opportunity this afternoon to introduce a wonderful colleague, Peter Hotez. But before I do, I wanted to announce to everyone that there will be a reception after the lecture today, sponsored by the FAES, which I think is a wonderful thing to allow us to have a time to interact with Peter, and it'll be in the library across the hall. So I hope many of you who have an opportunity can spend a few moments talking to Peter about some of the fascinating work that he'll present today. I'd like to start out by saying that Peter is probably one of the world's most renowned experts in tropical diseases. He's recognized internationally as a clinician and investigator of neglected tropical diseases and has spent a huge effort in vaccine development. He studies diseases that include hookworm, schistosomiasis, Chagas, leishmaniasis, and malaria, among others. And these diseases affect the lives of millions of individuals, and mainly children and adults worldwide. And obviously this is a critical public health importance and certainly related to the mission of NIH. He's president of the Sabin Vaccine Institute and leads the new Sabin Vaccine Development Program at Texas Children's Hospital and Baylor College of Medicine. He also serves as the founding dean of the new School of Tropical Medicine at Baylor and serves as chief of the new section of tropical diseases in the Baylor Department of Pediatrics. And he also holds an endowed chair at Texas Children's Hospital. Peter has been instrumental in creating the global network for neglected tropical diseases, which is an innovative partnership which is dedicated to controlling the spread of these neglected tropical diseases in developing nations. He got his PhD in biochemical parasitology from Rockefeller University and went on to get his medical degree from Cornell. He completed a pediatric residency at MGH and did postdoc training at Yale. And then he joined the Yale faculty as an assistant professor. He then achieved the associate professor before moving to George Washington University as professor and chair in 2000, where he has spent a number of years, and many of us as colleagues interacted with him at in that position down at George Washington. But he was wooed away by Baylor and is now settled in quite well. He's authored more than 240 original papers and has written textbooks. He's been invited speaker at many, many events, both nationally and internationally. And he is a member of the Institute of Medicine of the National Academy. He also serves as president of the American Society of Tropical Medicine and Hygiene and is the founding editor of PLOS, Neglected Tropical Diseases. So I'm very pleased and proud to have the opportunity to present Peter today. And he'll be speaking to us on the elimination of NTDs through anti-poverty vaccines. Peter, thank you very much for coming.
Dr. Zund, thank you so much for inviting me to speak to you this afternoon. It's always an honor to be here at NIH, and I never miss a chance to come up here to get intellectually rejuvenated. Something very special happened today with, I got to meet with the postdoctoral fellows from NIAID, and something that I can't remember when it's happened last. I actually got a free lunch and I try to impress upon the postdoctoral fellows how special this really is, and it could be years before this happens again in NIAID. But it's wonderful to be here. I've been in Houston 9 months, and one of the ways I was able to go was I convinced my wife that we actually moved in August, which is not the best time of year to move to Houston, but I tried to convince my wife that the summers in DC are just as bad as they are in Houston. Well, it's not quite. So and especially I was here, this is my second day I've been here, so the last 2 days have just been glorious weather, and so I know Dr. Zun arranged the weather, so thank you very much for that. So I'm going to be kind of giving you an overview talk about the problem of neglected tropical diseases and why you should care about these conditions, and so and give you some thought about where we're going in terms of thinking about a large global overview for eliminating many of the neglected tropical diseases. And this is the group that we're particularly concerned about, and this is a subject of concern at many of the institutes of NIH, not just the Fogarty International Center, but I think now this has kind of pervaded the entire culture of the institute, which is so nice to see this concept of the bottom billion, that there's 1.5 billion people in the world who live on no money, who live below the World Bank poverty, really officially live below the World Bank poverty figure of $1.25 a day, but I like to say for effect that they basically live on no money. And it's not just being dirt poor, but it's the fact that you live in a country, one of 58 nations, where there's sadly a certain inevitability about your poverty. It means your children and your children's children are destined to be poor. And it was with that in mind that many of you are aware of these goals that were established in the year 2000 by Kofi Annan, then Secretary General of the United Nations, to lift the bottom billion out of poverty and to practice sustainable poverty reduction. And what's interesting about these MDGs is we're now 12 years into them, and they're still around with us. Unlike so many United Nations documents that really have not withstood the test of time, oftentimes they're forgotten about as quickly as they're drafted. This one is stuck, and of relevance to the NIAID and other institutes is that there was one of the Millennium Development Goals, in addition to extreme poverty and hunger and gender equality, a lot of things to empower women, improve maternal health. There's one that specifically addresses infectious diseases to combat HIV/AIDS, malaria, and other diseases. And the NIH has really responded in a big way between certainly stepping up their research on AIDS and malaria, as well as a lot of other diseases. And the U.S. government in general has stepped up through PEPFAR, the President's Emergency Plan for AIDS Relief, which is now funded almost to $7 or $8 billion annually, and also the President's Malaria Initiative. But with the exception of the intramural and extramural research here at NIH, a lot of the major governments from the G8 and the G20 countries have really not done much about some of the other diseases. And the ones that we're going to be addressing today are what we call the neglected tropical diseases, or the NTDs. And it's a concept that we first wrote about in 2005 and 2006 to reflect the fact that there's a lot of commonalities between this group of predominantly parasitic and related infections that I'm going to list for you in order. So these are the most prevalent infections of poor people. Basically 1.4 billion people are infected. All of the bottom billion have one or more of these neglected tropical diseases. And I'm going to actually rank them for you in terms of the most common infections of poor people based on some of our studies, as well as some new information that Dr. Simon Brooker has developed from the London School of Hygiene and Tropical Medicine. So this is arguably the most common infection of poor people. Intestinal roundworm, ascariasis. A lot of these are wormy diseases. Hookworm infection, 690 million people. This little boy on the right has both hookworm and ascaris, and you can see he's got a big distended belly. He's also, and you can actually palpate the outline of worms in his intestinal tract from ascaris, but it's not just the fact that he's got a big distended belly. He's actually stunted for height and weight, and he also does very poorly on tests of cognition and intelligence, does poorly on IQ tests, and this, now we know, is because he's infected with one or more of these neglected tropical diseases, ascaris and hookworm. Here's whipworm infection, 520 million people. Some estimates suggest as many as 400 million. Others suggest as many as 600 million are infected with schistosomiasis. We're going to come back to that. 115 million people with filarial worms in their lymphatics and genitals, 40 million with trachoma. This is river blindness, liver fluke infection, leishmaniasis, and Chagas disease. Again, predominantly parasitic infections with the exception of trachoma, arguably the most common infections of the world's poor. And the consequences of this are, in addition to the health effects, we've now accumulated a lot of evidence to suggest that these are not only occurring in the setting of poverty, but the 17 major NTDs, as now defined by the World Health Organization, also cause poverty. And they can cause poverty because they reduce productive capacity. They make people too sick to go to work every day. And I'll show you an example of that. Or they're impairing intellectual and physical development in children. And I'll show you examples of that as well, or causing adverse pregnancy outcome. This concept that the NTDs not only occur in the setting of poverty, but they're a stealth cause of poverty and really are trapping the bottom billion in poverty. So how did they do that? Let me give you some examples. So here's a wormy curve that was generated in part by our group, but many other groups have shown the same thing. And it's basically showing that when you live in an endemic area, when you're looking at the intestinal worms, as well as some other types, that it's children that have the largest numbers of worms. So here I'm showing you age on the x-axis, number of worms on the y-axis, and there's a peak period of worminess during the life of an individual when you harbor larger numbers of worms than any other single time between the ages of 4 and 5 and 15. And what I find interesting is we really still don't know the basis of this. You know, the most common infection of children in the world, and we don't even really have our arms around why it is that children have larger numbers of worms than adults. Is it some type of acquired immunity that happens? Is it behavior? Is it exposure? We're still in the dark about that. So you get to this situation where you have this big distended belly and this little girl from Paraguay, and these are the worms from that one child. That you wouldn't see this in older adults and individuals, and we really don't know the basis of it. So the point being that the fact that children have so many worms, and especially the intestinal worms in this particular case, goes beyond just the immediate health effects that we've known about since medical school. And among them is this curious observation that the more worms you have, the greater your loss. Let me use this pointer instead. The greater your loss in intelligence, shown on the y-axis. And it's basically showing you, here you have negative worm burden, light worm burden, moderate, heavy, and this is loss of intelligence. So I like to tell my neurology colleagues or my psychiatry colleagues, here's the world's leading neuropsychiatric disturbance. And there really isn't a single published paper on the mechanism. I mean, presumably there's some kind of interplay with nutrition, but beyond that, it's really not well established. We know in the case of hookworm that iron deficiency may be related to it, and iron is a prosthetic group needed for the biosynthesis of dopaminergic neurons, but we really don't know the basis for it. We also don't know why it causes physical growth retardation as well. So here you have age on the x-axis, weight on the y-axis, and we're looking at a growth curve that you would see in any pediatrician's office, following children along the 3rd, the 10th, the 25th percentile. And here's a child with flatline growth, interval intervening to get rid of the worms, and then there's impressive catch-up growth. So here we are, maybe the leading growth disturbance, the leading cause of intellectual deficits and neuropsychiatric disturbance, and yet we really have very little information on mechanisms to account for this. So there's an enormous number of research problems that we could address with this. And now Hoyt Bleakley, who's an economist at the University of Chicago, has shown that fact on the right there, that chronic hookworm infection in childhood leads to a 40% reduction in future wage earning. Who knows why? Maybe through the intellectual and cognitive deficits. I think that's the most likely reason. But again, this recurring theme, NTDs actually causing poverty, not just occurring in the setting of poverty. A big impact on reproductive health. This is a study that we did with Simon Brooker at the London School of Hygiene and Tropical Medicine and Don Bundy at the World Bank, showing that there are an estimated 7 million pregnant women in sub-Saharan Africa with hookworm. 1/3 of pregnant women in sub-Saharan Africa with hookworm associated with reduced birth weight, increased perinatal child mortality, increased maternal mortality. And the big problem that you get with hookworm is profound anemia. That's a result of heavy infections that you can oftentimes see in pregnancy. So this is not only an infection during childhood, or as my obstetrics colleagues tell me who work in Africa, it's not because women at the time of delivery are bleeding more than women in the United States. It's just that they're starting out with 2 strikes against them because of severe hookworm infection and other neglected tropical diseases. And with that in mind, there's an increasingly recognition that these NTDs are not occurring in isolation, but there's comorbid effects with other infections. And a good example is coendemicity with malaria. And this is a study we did that Simon Brooker led, showing that if you look throughout sub-Saharan Africa, in the green are areas of Africa where you have both hookworm and malaria, predominantly falciparum malaria, occurring exactly at the same time. What this practically means, practically speaking, is that people are polyparasitized. They don't just have malaria or they don't just have hookworm, they often have both together. And on the right, you can see the consequences of having these co-infections is that there's a hemoglobin concentration on the y-axis, and I'm showing you some studies from western Kenya, central Kenya, northwest Tanzania, of what happens if you have neither infection, if you have malaria alone or hookworm alone or both together. And you can see the bottom really falls out when you have both infections together, this very profound anemia that you're seeing in children, that you're seeing in pregnant women as well. And so that really adds to it. And the term I used was the perfect storm of anemia that you're getting with malaria and hookworm co-infections. There's also increasingly recognition that these are important cofactors in Africa's AIDS epidemics. So there's a disease called what we used to call urinary tract schistosomiasis. It's caused by Schistosoma hematobium, and we've known for generations that the spine-shaped eggs, when they get into the bladder, will cause irritations, ulcers, and inflammation, so that they're responsible for blood appearing in the urine, a condition known as hematuria. So this is a common disease. We said maybe as many as 400 million people are infected, 90% in Africa, so that if you go into an endemic village and ask kids to void in a cup and then hold up their urine, as these kids are doing, a large percentage of children in sub-Saharan Africa have hematuria, have blood in their urine. But for decades, we've ignored the fact that we should be doing colposcopic examinations on the girls and women with this condition, and now there's recognition that up to 75% of them have those same eggs and lesions not only in their bladder, but on their cervix and uterus and lower genital tract. And it's a condition known as female urogenital schistosomiasis or female genital schistosomiasis. So I'm showing you, these are studies done by Jennifer Downs at Cornell, and Arian Jetland at University of Oslo has been looking at this throughout places like Mozambique, throughout Malawi, Tanzania, and where you can see these eggs causing granulomas and mucosal ulcerations on their cervix with contact bleeding, and it's associated with infertility, pain on sexual intercourse, but there's a lot of stigma and abdominal pain associated with it. And you can imagine that these granulomas are rich in eosinophils and CD4 positive cells and all of the things that Alan Scher and Tom Wynn have been working on for years and years. Well, that's all happening now in the female genital tract as well. And when people have bothered to look, there's now been 2 large studies published showing that in Zimbabwe that these lesions are associated with a threefold increase in HIV/AIDS horizontal transmission, and in Tanzania, a 4-fold increase in horizontal transmission. You can see a lot of geographic overlap between these 2 conditions, especially in southeastern Africa. The point being that this is not a rare condition. If you can do back of the envelope calculations to suggest that there may be as many as 100 million girls and women with female urogenital schistosomiasis. This could be the most common gynecologic problem in sub-Saharan Africa. I like to call it the most important infection or disease of girls and women you've never heard of. And it potentially is having a huge role in Africa's AIDS epidemic, but it's a neglected tropical disease and grossly unstudied. Another key point I'd like to make is that when we think about NTDs, we're very focused on sub-Saharan Africa and the poorest points of South Asia. But increasingly we recognize that poverty seems to be the overwhelming determinant of neglected tropical diseases. You show me poverty, I'll show you neglected tropical diseases. And an example of that, I want to take you to the Western Hemisphere in the Latin American and Caribbean region. That's what I mean by lack. If you look at the population of 578 million, basically 48 million live on less than $1 a day, and another 99 million in total live on less than $2 a day. It's a term that we sometimes call the bottom 100 million, living in our own backyard in the Western Hemisphere. And some studies we did a while back using the DALY, the Disability Adjusted Life Year, suggest that the DALYs lost from neglected tropical diseases is actually right up there with HIV/AIDS or tuberculosis, even more than malaria, so that there really is a fourth leg to this triumvirate of AIDS, TB, and malaria that we often say like a mantra. It really is AIDS, TB, malaria, and neglected tropical diseases. And here are the major neglected tropical diseases in the Latin American Caribbean region. Some are not unique to the Latin American region, things like hookworm, ascaris, and trichuris, or schistosomiasis or dengue, but Chagas disease is a unique one, and we're going to come back to that in a little bit. And you can see the percentage of poor people infected with these conditions is quite extraordinary, you know, between 10 and 85%. And again, they're not as ubiquitous in Latin America. They focus on those areas of poverty. So this is a map of poverty in Latin America, and to no one's surprise, it's in southern Mexico and Central America, very poor parts of Bolivia and the Gran Chaco between Bolivia, Paraguay, and northern Argentina, and regions of the Andes and in Colombia. And these are the regions where we need to look for neglected tropical diseases. What I'm showing you here is just a comparison of human development indices, because I like to make the point that poverty is not, and improper development is not exclusive to sub-Saharan Africa. Again, we have a lot in our own backyard, and we can look at the human development indices in some Latin American countries, and we can see that they're equivalent to many African countries as well. The other point about poverty in Latin America is even wealthy countries have intense pockets of poverty, and people are at risk for neglected tropical diseases. So 11 million people in Mexico live in extreme poverty, 10% of the population. 46% lives in some level of poverty, and it's particularly concentrated in places where we're working quite a bit now, in southern Mexico and up in the Yucatan as well. And if we can even take that one notch further, we have poor people in the United States. Who knew? So we have, the estimates are that 46 million Americans, people in the U.S. live below the poverty line, and Texas has the single largest number of cases of poverty of any state in the U.S. About 5 million people live in poverty, 1 in 5 Texans, but you get child poverty rates, that should be child poverty, approaches 50% in these counties along the southern border. So it's in South Texas, the counties in red, parts of the Panhandle, eastern Texas, high rates of poverty. Well, what about neglected tropical diseases there as well? So we recently came out with this paper looking at the neglected tropical diseases that are occurring in both Texas and Mexico, and this is our list of diseases that we're finding quite a bit of that I want to talk to you about a little bit today: Chagas disease, cysticercosis, dengue, leishmaniasis, and leprosy, and was the basis for which we actually made the decision to move our laboratories to Texas and to set up this new School of Tropical Medicine. So this is a risk assessment map of Chagas disease in Texas. My colleague Dr. Sarkar estimates that there may be as many as 267,000 cases of Chagas disease in Texas. Now you might say, well, that's all the immigration coming up over the border, but it turns out that it's now highly endemic among the dogs in South Texas. Around 11% of dogs are infected with Chagas disease in South Texas. We have the kissing bug vectors there. We have the poverty conditions. And so he's actually constructed a risk map of where Chagas disease will occur. So I think an important question that we want to answer over the coming months and years is what percentage of these 267,000 cases are actually as a result of endemic transmission of the disease within Texas. Because you have to be very careful when you talk about these diseases. There is this anti-immigrant mentality through many parts of our country, and every time I talk about it, the anti-immigrant blogs light up and they say, see, you should build the wall higher, and we're trying to make the case that you could build a wall as high as you want, but if there's transmission of these diseases, if we have poverty, if we have the right climate conditions, we have to really give serious thought to investigating these diseases, just like we would in any other developing country. We have to think about it here at home as well. Neurocysticercosis, now we're seeing in Texas, is one of our leading causes of epilepsy. And there was a nice paper out in Discover magazine where they interviewed Dr. Ted Nash about the problem of cysticercosis here, and it's clearly a huge problem, and we're seeing this disease regularly show up in our doorstep. Another one that we're quite interested in that we're seeing in Texas, but elsewhere in the American South, is one that not many people give a lot of thought to. It's called toxocariasis. And it's a disease that's transmitted from dogs. It's basically like a dog ascarid worms, where you swallow the eggs, and then the larvae migrate through the tissues where they'll cause inflammation. So the full-blown syndrome is fairly rare. It's called visceral larva migrans. But we're seeing a form of the disease that we're calling covert toxocariasis that's linked with eosinophilia, and we also see a lot of wheezing. And there's been studies in Europe actually showing that Toxocara larvae, these parasitic nematode larvae, are an occult cause of asthma in Europe. The question is now, the CDC has shown a 21% seroprevalence among African Americans, with poverty being the overarching risk factor. What's the role of Toxocara infection, covert Toxocara infection, in promoting asthma and developmental delays? And so there's a lot of hypothesis generation going on, and we think these will be fertile grounds for study. We have dengue. So this is, we know we've had dengue in western Florida caused by dengue type 1. We've known we've had dengue type 2 in Brownsville, Texas. Christy Murray, who's a new faculty member with us, has been looking retrospectively at stored serosamples for the past 10 years, and she's uncovered pretty good evidence, and this is a paper now under review, that we've had dengue outbreaks in Houston during May and June every year, possibly for the last 9 years. So we have the point being transmission. Aedes aegypti mosquitoes are here. There's also Aedes albopictus. So there's dengue as well. So when you look at it all, Chagas disease, cysticercosis, dengue, Toxocara infection, there is, and these are not even rare diseases. I'm not even sure these would qualify for diseases under the Orphan Drug Act by the Food and Drug Administration. These are common diseases, and I often like to say if these were occurring among wealthy people in the suburbs, we would never tolerate it, but because they're attacking poor people living in extreme poverty, especially people of color, there really are forgotten diseases among forgotten people. So one of the things that we've done now is at the Ben Taub General Hospital, which is a public hospital at Texas Medical Center. It's an extraordinarily well-run public hospital. We've created a tropical medicine clinic where patients from the area come every Friday morning, and the things that we're seeing on a weekly basis, leishmaniasis, Chagas disease, cysticercosis regularly, with some of it from transmission within Texas, we think is going to be very interesting. And I think this is, I've been talking to people at NIAID today, that we think this is a great opportunity for fellows at NIAID to come to Houston and see some of this pathology. We would love to have you. Now we've created a new diploma in tropical medicine, because with bringing the Sabin Vaccine Institute that I'll talk to you about in a minute, as well as this endemic tropical diseases, we've now created this new National School of Tropical Medicine, which is giving its first diploma in tropical medicine that just started last week. And so we're hoping to offer it on a twice-a-year basis, once in the summer and once again in the winter. So what I'm showing you is Houston. There's downtown Houston, and this is not another city. This is the Texas Medical Center, which some people say is the largest medical center in the world. It's really a medical city of 100,000, nicely 96,000 employees. that has its own police force. It's just an amazing place, and it's a great opportunity to now take all of that and really concentrate global health activities in one area through this National School of Tropical Medicine, very much modeled on the Liverpool or London School of Hygiene and Tropical Medicine, but we've adopted a New World theme for our logo rather than the Egyptian theme of the Liverpool School or the London School. This is the Mayan jaguar, the protector with the cacao tree. This is something that we've just started. All right, so now that's kind of the overview of the burden of disease caused by neglected tropical diseases. I want to kind of update you on what's happening in terms of global control efforts to either control or eliminate these diseases. And the first thing I want to tell you about is this concept of mass drug administration, which is blanketing large populations with a single dose annually of a drug to reduce the burden of these diseases. And this concept was not discovered by Stephen Hawking, but some people credit his father with potentially discovering this. His name is Frank Hawking, who was working with a drug called diethylcarbamazine citrate, studying a disease that's heavily investigated here by Amy Cleon and Tom Nutman, lymphatic filariasis, where he was able to show that if he could give a single dose of diethylcarbamazine citrate, abbreviated DEC, it actually reduced the number of circulating microfilaria, which is required for transmission of the infection when it's picked up by mosquitoes, and was able to provide evidence that you could, in villages in and around Recife in Brazil, that you could actually eliminate the disease as a public health problem by treating entire villages at the same time, and even took it a step further, showing you could mix the medicine up in the cooking salt. So if people are salting their foods by cooking, they could dose themselves with the medicine. And he wrote about this in the Bulletin of the World Health Organization in 1967, which got read by a Chinese parasitologist during the Cultural Revolution. And what they did was, this actually went to the attention of Chairman Mao, and the Chinese scaled it up as only the Chinese could by medicating the entire salt supply of eastern China with diethylcarbamazine citrate, thereby becoming the first country to eliminate the disease as a public health problem, again, through this concept of mass drug administration. And so the World Health Organization began looking at how you could franchise this. And they worked very closely with another very important person who was at NIAID, Eric Otteson. And Tom Nutman and Amy Cleon have been heavily involved in these studies, showing that through mass drug administration you could actually reduce the prevalence of these diseases, either through once-a-year diethylcarbamazine citrate or through medicating the salt. We can't use diethylcarbamazine citrate in Africa because it causes a severe toxic reaction known as the Mazadi reaction in patients with river blindness, which is co-endemic. But a Merck company came through by donating ivermectin for that purpose. And so you have two drugs now, diethylcarbamazine citrate, where a box of 1,000 tablets costs $4. So we're talking about 0.4 cents a tablet, whereas in Africa we're using ivermectin, which is free. So, so you're basically factoring out the cost of the drugs. And what was very exciting is the CEO of Pfizer said, well, if the CEO of Merck and company is going to donate something, I better donate something too. And he donates Zithromax, which I'm sure many of you have taken in your Z-Packs sometime in the past year for your upper respiratory infection. It works for trachoma, and this is a result of praziquantel administration in Egypt. So a few years back, we undertook this exercise with the WHO and with David Molyneux at the Liverpool School of Tropical Medicine and Alan Fennick at Imperial College London, along with some others, including Eric Otteson, to sketch out a map of the world for where these neglected tropical diseases, these 7 most common ones that could be targeted through mass drug administration, meaning ascariasis, trichuriasis, hookworm, schistosomiasis, lymphatic filariasis, river blindness, and trachoma. And because there had been enormous successes through the global program to eliminate lymphatic filariasis through mass drug administration, reaching about 50% of the populations at risk, but we did much worse with targeting the intestinal worms with benzimidazole and helminthics like albendazole or mebendazole or praziquantel coverage. But you can see in a place like Sudan, where you have all 7 neglected tropical diseases, what you're seeing are that people are polyparasitized. They don't just have schistosomiasis, they may may have schistosomiasis and river blindness, or they may have hookworm, ascaris, and schistosomiasis. So the concept came that perhaps we should bundle it all in a package, which we sometimes call the rapid impact package. And the idea is to combine albendazole or mebendazole, which will target especially the intestinal worms, the ethylcarbamazine citrate or ivermectin to target the filarial worms, praziquantel for schistosomiasis, and then zithromax for trachoma, with safety data now to support that you could give albendazole, ivermectin, praziquantel at the same time. And you could target all 7 neglected tropical diseases at once: ascariasis, trichuriasis, hookworm, lymphatic filariasis, onchocerciasis, schistosomiasis, and trachoma. And we're now providing proof of concept that we can do this for as little as 50 cents a person per year. So think about taking on these diseases, the most common infections of humankind, and knocking them down, in some cases actually eliminating lymphatic filariasis, eliminating river blindness, eliminating trachoma for 50 cents a person per year. So we wrote those papers in 2005, 2006, and at that time, Eric Otteson, together and his colleagues, projected that this integration of combining mass drug administration would lead to a 26 to 47% reduction in cost savings. And sure enough, the Carter Center, Frank Richards and others recently published that you got about a 41% reduction in cost by combining these medicines into a package that can be given closely spaced apart. And USAID has now backed this concept really nicely. It began in the Bush administration and has expanded in the Obama administration, where I'm showing you the funding in millions of dollars. This is the funding for integrated NTD control since 2006, and excuse me, getting up to $89 million annually. Now that may not sound like a lot when you're thinking about PEPFAR dollars of $8 billion, but because the costs are so modest, that we can make a very big impact on these diseases through national programs of integrated neglected tropical disease control. We still have a ways to go. One of the big problems that we face in doing this is, in order to develop algorithms for treating with the different medicines, we need to have good mapping data on where these diseases are. And this is just to show you where we're at. This is a paper done by Jörg Utzinger from the Swiss Tropical Institute, just looking at schistosomiasis. And you see this big hole over here in the Democratic Republic of Congo. You remember the Joseph Conrad book, Heart of Darkness? Well, it's also darkness in terms of data collection. So we have big gaps in our data. It's not that schistosomiasis is not there, it's just that nobody's looking because of the complexities of working in conflict and post-conflict countries. Same with Central African Republic as well. We're also working on version 2.0 now of integrated NTD control, which is that we need to really start thinking about how we're going to link this with the President's Malaria Initiative and with PEPFAR, especially when you're seeing the impact of praziquantel administration potentially because of lowering rates of schistosomiasis on reducing the prevalence of that disease. If that can reduce rates of female urogenital schistosomiasis, would we have a big impact? Should we be including NTD control in PEPFAR? Should we be including NTD control in the President's Malaria Initiative? We think we should. It's just a matter of figuring out how to operationalize it all. So this is going to be an important new trend in the coming year. Now, those of you who are scientists or might be thinking, my God, when we talk about treating literally hundreds of millions of people, aspiring to get all of the bottom billion on these anhelminthic drugs and other NTD drugs, aren't we concerned about emerging drug resistance? And the answer so far is there's not a lot of evidence for emerging drug resistance against these NTD pathogens. On the other hand, not a lot of people are looking as well, and our status of biomarkers for these diseases is quite inadequate. But what we are concerned about is, for instance, one of them, which is single-dose mebendazole. What I'm showing you here is a forest plot which is showing the improvement effects of anemia on hookworm when you use albendazole, because it's to the right of the middle line here. But it's showing that mebendazole is having no impact. In fact, a new meta-analysis published in JAMA, I guess it's not so new anymore, about 4 years old, has shown a 15% cure rate for mebendazole. The issue is, sorry, my mouth's getting a little dry, talking fast. The issue is we don't know if this is new and whether it's really truly emerging drug resistance or the medicines haven't worked well in the beginning. So that single-dose mebendazole just may not be a good medicine for hookworm, but it points to the fact that it's, I think, as much as of an advocate as I've been on promoting mass drug administration and integrated NTD control, there's going to be there's an extreme importance to not stop innovation, not stop research and development efforts, and that's been a very difficult message to persuade, whether it's Congress or other global health policy people. I mean, the Gates Foundation gets it, but not too many others, that you can't, it's not either or, it's not just mass drug administration. You have to continually invest in research and development for new and better control tools. And this is a paper that I wrote a couple of years ago, actually 2011, saying that to offer the concept that for many of the neglected tropical diseases, we need to think about developing new vaccines. It's not enough just to give the medicines, but we need to think about vaccinating in order to prevent reinfection as a potential elimination strategy. And to be as provocative as I could, to get people to notice it, we call them the anti-poverty vaccines, because they'd be vaccines that have not only an impact on improving global health, but improving educational outcomes and improving poverty outcomes for all the reasons that I showed you about in the beginning. So back in August, the best time of year to move to Houston, we moved our laboratories to this building over here, this enormous research tower. It's called the Feigen Center, named after the great pediatrician Ralph Feigen at Texas Children's Hospital, where this is our portfolio of vaccines that we now have under development. And I don't have too much time to go into detail, but I'll tell you about a few of these vaccines briefly. So we have a human hookworm vaccine that's has a component now in phase 1 clinical trials, and that's being led by David Demert, who was formerly at NIAID and is now linked with the Sabin Vaccine Institute. Schistosomiasis vaccine that's complete in manufacture and we're waiting to start clinical trials. A new Chagas vaccine, as well as a brand new SARS vaccine. So the hookworm vaccine is supported by the Bill and Melinda Gates Foundation and the Dutch Ministry of Foreign Affairs. The schistosomiasis vaccine is supported partly by NIAID, but we've got together a consortium of private individuals such as Gary Michelson and the Blavatnik Charitable Foundation and Mort Hyman. And the Chagas vaccine is supported by the Southwest Electric Energy Medical Research Institute, a local institute in Houston, together with the Carlos Slim Institute of Health, and NIAID is supporting the new SARS vaccines. And we're doing this through the Sabin Vaccine Institute, which is a type of organization that some of you might not be familiar with. And then it's a type of organization which is sometimes known as a PDP, a Product Development Partnership, which are essentially nonprofit organizations that use industry practices to develop stuff, to develop products, whether it's diagnostics, therapeutic products, which basically means small molecule drugs, microbiocides, or vaccines to target different neglected diseases, defined broadly to include AIDS, TB, malaria, NTDs, and diarrheal and respiratory diseases. And our sweet spot are vaccines, crossing over here with NTDs, which is where our major focus is currently. So our hookworm vaccine, our goal is to reduce 80% of moderate and heavy hookworm infections, which will cause worm burden reduction, egg reduction, reduced intestinal blood loss, reductions in anemia and malnutrition, and thereby be dose sparing as well, drug sparing, to prevent ongoing use of the drug to delay or forestall emerging drug resistance. Many of you may not be familiar with hookworms. The major species responsible for 85% of the global hookworm infections is Necator americanus, which is a helminth parasite about 1 centimeter long that fastens deep into the intestinal mucosa and then causes blood loss, and then the blood comes out the other end. So it can produce profound anemia and a kwashiorkor-like illness. We've spent now decades trying to develop strategies for vaccinating. We don't have, and we could do a whole hour just on how you make a vaccine for something like this. Remember, this is a worm, an animal about 1 centimeter in length. But we've really been focusing in collaboration with Dr. Alex Lucas and Jeff Bethany and others on the gut of the worm. And what happens is, as the worm feeds on blood, we've been able to show that the worm breaks open the red blood cell and is particularly focused on ingesting hemoglobin. I know you're not used to looking at CAT scans of worms, but here's a longitudinal section of a hookworm that's been probed with an antibody against enzymes used to degrade hemoglobin. You can see that along the brush border membrane of the gut, possibly tethered by putative GPI anchor sequences, are enzymes involved in hemoglobin digestion, including an aspartic protease known as APR1, a cysteine protease known as CP3, and a metalloprotease that breaks down the constituent hemoglobin into smaller bits that are then absorbed by an amino acid transporter, and then the parasite has to do something with heme. Blood-feeding parasites like malaria hate heme. In the case of the malaria parasites, it polymerizes it into an inert pigment known as hemozoin. In the case of hookworms, they deal with heme by binding it up through a specially modified glutathione S transferase, which forms a homodimer and has a low peroxidase function, but is specifically bound to heme. And so through the years, we've identified 2 molecules for purposes of vaccine development. One is that glutathione S transferase to elicit neutralizing antibodies. The other one here that looks like the continent of Australia is an aspartic protease, but the protease has been modified through site-directed mutagenesis to modify 2 aspartic residues to alanines so that it is catalytically inactive, yet keeps its tertiary conformation. We've been able to show that this will elicit high levels of antibody. The antibodies are then ingested by the worm. The antibody neutralizes blood feeding and then inhibits the ability of the worm to survive. So it's a very novel mechanism of how you use the immune response to attack an animal. And here is our clinical development plan, which includes phase 1 studies in adults for each of the antigens, then combining the 2 and ultimately establishing proof of concept for protection. We are now also seriously looking at the possibility of doing some challenge studies in vaccinated human volunteers to challenge them with hookworm larvae, and this was actually pioneered back in the 1980s at NIAID, this concept of doing self-infections with hookworm larvae, pioneered by Eric Otteson, working with Frank Neva, and Tom Nutman presumably was also involved, of actually placing hookworm larvae on people to look at self-infections. We've been asked to really think about developing a challenge model in order to make all of this run faster. And our modelers that we've been working with have been able to identify some real advantages of using a hookworm vaccine rather than getting this rebound reinfection that we're seeing with albendazole or other anhelminthic drugs, that over time this could lead to elimination of the disease as a public health problem. And we're going to come back to this. What do we need to eliminate the 17 leading neglected tropical diseases as defined by WHO? Again, using the mass drug administration approaches and then folding in new tools, new control tools such as vaccines, so that in the case of lymphatic filariasis, while we might be able to eliminate the infection with mass drug administration alone, our thinking is that for hookworm, the MDA, the mass drug administration by itself will not be sufficient. We'll need to fold in vaccines. And then there's some nice studies that Bruce Lee and his colleagues have done at the University of Pittsburgh. Chris Bacon is part of that group to do some modeling looking at the impact of DALI reduction over a period of 20 years with drug treatment alone versus adding the vaccine and having a big improvement. Alex Lucas, who was with us and is now has his own group at James Cook University in Cairns, has been working also with Jeff Bethany to take advantage of completed genome projects supported by NIAID to identify this very exciting tetraspan and surface antigen on schistosomes, which when you formulate it with an adjuvant, such as alum or a synthetic lipid A, you can get very high levels of antibody that seem to result in high levels of worm killing of around 50%. And Alex has done some nice RNAi studies. You could use, we can't get RNAi to work in parasitic nematodes, or at least in hookworms, but he's been very successful getting it to work in schistosomes and showing he could ablate the biogenesis of the schistosome tegument using antisense RNA to this molecule. And now we can show high levels of protection in laboratory mice. So we've just through a number of different sources of support, including NIAID support, we've now are finished manufacture of this and are advancing it to clinical trials. Our global access plan for how we're going to get these vaccines disseminated to the world's poorest people has a heavy reliance on working with developing country manufacturers. So one of the great things about, if you look at the 3 countries in Latin America where vaccines are made, which is Brazil, Cuba, and Mexico, is their public sector. They're actually owned by the federal government, oftentimes an arm of the Ministry of Health. In the case of Fiocruz or Butantan is owned by the state of Sao Paulo, and their goal is public health rather than funding money for their shareholders per se. And we're looking now at developing the manufacturing on a large scale the hookworm vaccine jointly with Fiocruz and their biologics manufacturing arm known as Biomanguinhos. The old campus name of Fiocruz was known as Manguinhos. And for the schistosomiasis vaccine, doing that jointly with Instituto Butantan, which has great capability. And then what's impressive about Brazil, it has great biotechnology prowess, but also endemic tropical diseases. And we do the clinical testing. This is led by David Demertz and Jeff Bethany's group out in rural Minas Gerais state, where the vaccines are being tested. And here you're getting incredible prevalence of hookworm, 68%, schistosomiasis, 45%. So Brazil is an example of what we sometimes like to call an innovative developing country, an IDC, a country with great pockets of poverty, and yet they have great ability to produce biotechnology products. Mexico is another example, and you could think of others in Asia, such as India and China that makes vaccines, or Indonesia that makes vaccines, or Thailand and Vietnam make vaccines. And right now, as far as we know, there's only a single sub-Saharan African country that makes vaccines. That's Senegal, that produces a yellow fever vaccine. With all of the Chagas disease that we're now seeing, we've got very interested in this problem. And again, through an NIAID funded genome project, a number of promising antigens have been developed, including one developed in Eric Dumontil's laboratory, 2 antigens known as TC24 and TSA1. And now we're looking at scaling up these antigens for a Chagas vaccine that I'll tell you about. So Chagas vaccine, I'm relatively new to the field. We've only been working in it for a couple of years. And I've just gotten fascinated by this disease because of its high prevalence, with some estimates saying there's an estimated 10 million cases of Chagas disease, but I don't think anybody really knows. Some people say there may be 6 million cases in Mexico alone, including the 200,000 plus cases in Texas and the United States. And of those 10 million people, roughly 2 to 3 million, 20 to 30% are going to go on to develop Chagas cardiomyopathy, which is linked with arrhythmias, heart failure, dilated cardiomyopathy, and sudden death. The current numbers are that 20,000 deaths occur annually from this condition, but some people feel the numbers are a lot higher. But it's also emerged as an important maternal health problem as well. New estimates suggest 11% of pregnant women in Latin America are seropositive for Chagas disease, and there's maternal transmission of around 5 to 10% and transfusion-associated cases. So based on all of that, you know, back when I was at Yale, I had a pediatric AIDS clinic and thought a lot about pediatric AIDS, and I said, you know, the maternal child transmission, the transfusion association diseases, it's a chronic infections. You know, with Chagas disease, the medicines do not work very well, especially as soon as you develop heart symptoms. High toxicity, often causing patients to abandon treatment. They're contraindicated in pregnancy. You know, I said, this reminds me of when back in the 1990s when I had a pediatric AIDS clinic. It reminds me of the early years of the AIDS epidemic. So I thought I would write it, Chagas disease, new HIV/AIDS in the Americas. And I since realized that you can't compare anything to HIV/AIDS without creating a firestorm. So this just came out last week. And we've now we actually have this ability to do article-specific metrics for our plus NTD papers. And the most we've ever had was around 20,000 hits, downloads of a manuscript, which occurred over a period of 4 or 5 years. We've now had 30,000 downloads in the first week of publishing this paper. That's the good news, but unfortunately a lot of the news bureaus kind of took it over the top and were a little too literal in what we meant by HIV/AIDS, thinking that there must be person-to-person transmission, which there's not, which is it's about to overtake New York City and Boston, which it's not. So I learned an important lesson of how you use the term HIV/AIDS in any kind of comparison. But I don't want to take away from the fact that this is a horrific, horrific problem of 10 million cases. And so when I called it the new American HIV/AIDS, what I really meant is that this is a problem of comparable magnitude in the poorest parts of Latin America and has a lot of similarities with respect to its chronicity, maternal-child impact, and transfusion-associated cases. So we're very interested in developing this is a therapeutic vaccine. And the idea here is because there's a lack of efficacy for current medications in seropositive adults, the drugs oftentimes do not actually eradicate the parasite nor induce seroreversion. It has a lot of intolerance and unacceptable side effects, so roughly 12 to 18% of patients have to suspend their therapy. It causes weight loss and urticaria. It's really our horrific drugs. They're contraindicated in pregnancy and quite expensive. We're looking at the possibility as a target product profile of a vaccine with these two antigens together with an adjuvant alum plus the synthetic lipid A, E6020, as a therapeutic vaccine to take people who have seroconverted and prevent or delay the onset of Chagasic cardiomyopathy, which would be possibly targeted for adults and children. And we're doing this through support from the Carlos Slim Institute of Health and the Southwest Electric Energy Medical Research Institute. And the idea would be that we're doing this just like we're doing the Hookworm vaccine and the Schisto vaccine jointly with the Brazilians. We're now doing the Chagas vaccine jointly with the Mexicans, including the public sector vaccine manufacturers in Mexico known as Bearmex. So this is an exciting project. One of the things that we're having we're going to struggle with is there's strong evidence that the vaccine protection appears to be dependent on CD8 positive cells and cytotoxic T lymphocytes, not antibodies. So that the problem that we're going to face is how do you produce strong CTL responses in CD8 positive cells to these recombinant protein antigens. Producing high levels of interferon gamma and CD8 positive responses, which has been shown as a DNA vaccine, will reduce parasitemia, decrease T. cruzi parasite burden in the heart, increase survival and decrease inflammation. How do we reproduce those effects? Because we think the DNA vaccines often have not shown a great track record. So we're in humans. So we're looking at the possibility now of co-formulating our vaccine antigens into nanoparticles. So one of the things that we learned coming to Houston is that Rice University has got some of the best nanoparticle institutes in the country, and they're our neighbors. So we're working very closely with them. We've recruited a man named Michael Heffernan who's been looking at now repackaging our vaccines in nanoparticles for delivery as a Chagas vaccine. So I'm going to stop here, and I think that is where we're, you know, the big picture is that the Bill and Melinda Gates Foundation launched this audacious goal for, they used a different E word, they used the other E word, the eradicate word for malaria. We're going to use elimination of the high prevalence neglected tropical diseases, and we think that's going to be through urgent needs to scale up mass drug administration, getting other countries involved, and scaling this up to cover all of the bottom billion. But again, continue to make new drugs, diagnostics, insecticides, and vaccines. And today we've spoken a little bit about the vaccine piece, but the others are, of course, equally important. So thank you to NIAID and to NIH. And to Fogarty International Center, where I'm a member of the advisory council. So NIAID has provided a decade of support for the human vaccine, for human hookworm vaccine. They've supported the Schistosomiasis vaccine. And again, 30 years of mentoring ever since my MD PhD days. I've always come here for whenever I've had tough intellectual scientific problems, as well as emotional support when you're doing something like making vaccines for neglected tropical diseases. This has always been a source of great comfort and learning, and I wanted to thank you for that opportunity. Stop there. I can take a few questions. Yes. Hi.
Hi. Thank you for that great talk. You referred to how most individuals with these infections are polyparasitized, and you talked about bundling therapeutics for these individuals. It's premature probably to talk about bundling vaccines for these individuals, but I wonder how much forethought is put into vaccine development when considering that some of these individuals with all these different types of parasitic infections as well as viral infections and bacterial infections may have immunologic interaction between the pathogens, and how that might affect vaccine development.
So it's a great question. I can't give full justice to it now, but because of the time. But one of the things, we absolutely agree with you. We're looking at bundling, for instance. You know, if I had a crystal ball, I'd love to see the day where you could bundle hookworm and schistosomiasis vaccines because they're co-endemic. The others, when you think about the time of delivery, we're most likely going to want to integrate our neglected tropical disease vaccines in a health system where kids are getting vaccinated at around the same time. So the most likely one would be maybe that last EPI vaccine, the expanded program of immunization, giving it towards the end of the first year of life when they're getting measles vaccines, for instance. We think potentially that could be a very good time to do it rather than trying to reinvent the health system. And then you've got to do all the immunological interference studies that go along with that. So we've got our work ahead of us. Yes, Tom?
It's a really nice talk, Peter. I'm curious how you're getting around the complications of the urticaria that you saw in the first trials with the hookworm vaccine, you know, because of the induction of IgE and mechanisms of resistance. How are you getting around that?
Thanks for that question. So I didn't have time to go into it, but this is not our first generation recombinant protein hookworm vaccine. The first one is a one that we worked on in our lab. It was an antigen first discovered by John Hawden, who was at the time a postdoc in my lab, and we had worked on for 25 years, and we watched it crash and burn one day all suddenly when we immunized people in the endemic area, and 3 of them developed generalized urticaria. And it turned out that about 30% of the endemic population in Brazil, as they were living in the endemic area, developed IgE antibodies to those antigens naturally. And then when we administered it as a vaccine, we were cross-linking that IgE on their mast cells and causing generalized urticaria. So that's given us a very sobering lesson for how to advance helminth vaccines into clinical development. So what we do now is we look for levels of pre-vaccination IgE in endemic communities. And the reason, part of the selection criteria now for all of our helminth antigens is no IgE in the endemic area. So there doesn't appear to be IgE in GST1 and APR1, and sure enough, so far we've had no problems with GST with respect to urticaria. The harder question is, why do some antigens have high levels of IgE in the endemic area and others have none? And it's interesting that the ones we've looked at that have IgE are mostly the third stage infective larval antigens, but the adult hookworm antigens don't seem to have that problem. So we're really trying to sort that out. Why? And the same with TSP2 for schisto. IgE does not appear to be a problem, but Alex Lucas is now looking at some protein arrays and making an IgE chip to really look at all the different schistosome antigens, and some of them that are being talked about do have high levels of IgE, and the question is, will that be a showstopper for advancing them into clinical development?
Peter, I just want to thank you again for a wonderful talk,
Thank you.
Thank you for coming. And to remind the audience, we will have a reception sponsored by FAES in the library, so please join us and get all your questions that you wouldn't ask now to Peter later. So thank you once again, Peter, for coming.
Thank you again, Kathy.